there's no presentation pending. Hello guys, my name is Melih, and in this presentation, I'll be talking about designing a functional lipid nanoparticle for targeted alveolar drug delivery. So to just briefly go through the alveoli. The alveoli is made of mainly two types of cells and some immuno cells. Type 1 and type 2 epithelial cells make up the majority of the cells in the alveoli. What you don't see in the image is that alveolar macrophages. They're essentially the first immune response for anything that enters the alveoli, whether that's pathogenic or foreign particles. Type 1 cells essentially make most of the surface area, whereas type 2 cells usually have regulatory functions. One of the things that type 2 cells are heavily involved in regulating is the alveolar surfactant. So the image there shows a very thin layer on the top covering the alveolar epithelium. And that is essentially the alveolar surfactant, and it's a very thin aqueous layer of phospholipids and surfactant proteins. Together, they actually make sure that the lungs don't collapse, ensure that the alveoli remains elastic, and the airspace remains dry, and the cells remain humidified. What makes the alveoli special is the shared basement membrane with the capillary bed, essentially. There's a very thin diffusion distance between the alveolar sac and the blood vessels, which means that gas exchange can happen very rapidly and efficiently. So how do we achieve deep alveolar deposition? So one of the main criteria is to have formulations that are smaller than 3 microns in diameter. Particles and formulations larger than this will deposit at the higher branches of the airways, whether that's the trachea or the bronchi or the bronchioles, but will definitely not get to the alveoli. So first thing first, get particles that are smaller than 3 microns to make sure they get to the alveoli. Second thing is essentially to avoid the macrophages that we talked about. So although to deposit to the lungs, you need to have smaller than 3 microns. To easily evade the macrophages, you want to go even further smaller. So macrophages, they clear out everything pretty well, except things that are smaller than 200 nanometers, it starts to struggle at clearing. So having particles smaller than 200 nanometers is going to give it some immunoevasive properties. Lastly, the nanoparticle needs to be uptaken by your cells, so the targeting of your lipid nanoparticle needs to work. So I'll just quickly go into the aims and the hypothesis of this part of my project. So first, I need to produce a lipid nanoparticle with surface modifications and cell targeting properties. Once I achieve that, I need to quantify how these lipid nanoparticles are interacting with my cells. And I can do that by employing things such as confocal microscopy. I can really look closely at as to where the nanoparticles are and if they're being internalized how. And lastly, I want to integrate both of these things into a lung on a chip model. So this is exposing my optimized lipid nanoparticles into a microfluidic lung on a chip model. So essentially, I have two microfluidic devices, one to make the lipid nanoparticles and one to expose the nanoparticles onto the alveolar cells in a chip that mimics human lungs. The earlier stages of my PhD was very much looking at how we prototype microfluidic devices. The very first things I had to learn was how to design in 3D and learn parametric modeling. But this was very important because for me to be able to rapidly prototype things, I need to be able to rapidly design them. When I first came into the lab, there were a few techniques that we were using. The main one was photolithography. Some people spoke about this earlier today, and that's normal because it is the gold standard. It's got nanoscale resolution, can produce very fine channels, and it's a very well-established process. The things I don't like about it is that it requires a clean room, which means that I have to get out of the lab. And that's time consuming. But on top of that, you need to have specialist equipment and you need to pay for the clean room, ensure that everyone using the clean room has been trained appropriately. So it's not quite rapid prototyping, but just prototyping if you're using photolithography. The method that our lab was able to tackle this was they actually bought a xerography machine. Now, a xerography machine, anyone can buy this. People commercially use this to make stickers. But if you get fine enough blades, you can actually cut out geometries that are good enough to run microfluidic experiments. And this directly tackled all the problems of photolithography. It was very low cost per mask, and you were able to produce microfluidic chips and molds very quickly. The problem with the xerography is that it's got very low dimensional accuracy. So your future smaller than 100 microns, you can kind of forget about them. You won't be able to draw them appropriately. And on top of that, wherever it cuts, there is a very rough surface, which means that if you're trying to run microfluidic experiments that require laminar flow, for example, those rough surfaces will impede on those experiments. 3D printer is where essentially my savior is. I saw a 3D printer in the corner of the room that no one was really using, and I decided to put it to test. This was the reason it was purchased. I later found out, obviously, but it was essentially something that sits in the Goldilocks zone. It was very rapid, so we could easily directly go from designing something on a 3D CAD software to fabricating the molds directly on a resin 3D printer. And this allowed for rapid one-step microfabrication directly from CAD softwares, and we were able to issue features down to 10 to 20 microns, although I do just stay slightly higher than that. But the benefit is that the surface roughness is very minimal, and it kind of is the best of both worlds, and I can do it in the lab without leaving. The material costs are slightly higher than geography, but it's nowhere near as much as photolithography. Quickly moving on, how do we prepare our molds? Now that photolithography, no one's really doing it in our lab because we have to go out of the lab to get really accurate molds. I'm sure when we get to the end stages, we'll have to make some more accurate molds. But for the time being, the main method that everyone's using is the 3D printing method. So once you design your microfluidic geometry, you can see a green design on top. That is actually for lipid nanoparticle synthesis. It's a flow focusing device or a football field, as my sister told me yesterday. But that's something that you can design, and you will directly be able to print that exact mold on a 3D printer as it's seen there. Once you pick your choice, whether that's xerography or 3D printing, you can go ahead and follow on the steps. We talked a lot about nanoparticle formation, but nothing about what is actually happening. So lipid nanoparticles actually form via self-assembly on the laminar flow in the flow focusing chip that I discussed about earlier. And to give a brief background as to what we're doing here, on the right-hand side, you can see a lipid cocktail that I use to make my lipid nanoparticles. So these lipids are dissolved in isopropanol, an organic solvent, and this organic solvent is rapidly and in a very controlled laminar flow methodology. I'm kind of waffling the words there, but in a very, very controlled way mixed with the water, which triggers a form of nucleation. And this is not the traditional kind of nucleation that everyone talks about. In this case, you have lipids, which means you have a hydrophobic and a hydrophilic head and a tail. So these lipids naturally turning around to their respective favored side starts a point of nucleation, and nanoparticles quickly grow on from that first point. By doing so, the lipid nanoparticle slowly forms a little vesicle, and as it's closing up, it's encapsulating all of the content that it's surrounded by. And essentially, we can do this in a very controlled way. Particle size and encapsulation efficiencies are mostly down to how your flow rate ratio settings and your total flow rate ratio are. And lipid composition, of course, has a massive impact on your lipid nanoparticle size at the end. Just to quickly touch up on the lipid nanoparticles that the lipids that we are using. So the main lipid that we're using is DPPC, which makes up about 68% of our mixture. And we use DPPC because it is the primary phospholipid in pulmonary surfactant in humans. On top of this, we use lipids such as POPC. This essentially makes DPPC's properties a little bit less rigid. If it's too rigid, we will not be able to aerosolize it later on. It will become too fragile. And essentially introduces some fluidity. POPG has a very, very important role, and one of the roles is it makes the lipid nanoparticles negative. And due to the sheer size of our lipid nanoparticles, they're being very small, it being negative means that they're repelling each other, and it prevents aggregation in solution. More importantly, it does reduce the transient temperature or the transmission temperature, which allows the experiments to be conducted in room temperature as opposed to going to temperatures like 55, 56, 60 degrees Celsius, which is a much less friendly environment. Now, we did directly go and expose ourselves to some of these lipid nanoparticles. We've exposed them to healthy cell models and also some lung cancer models like A549s. And we had some preliminary early cell exposure results from this. So at the top, you can kind of see the Halley cells. The first box essentially highlights the first box essentially highlights the nuclei, so nucleic dye that we have highlighted. nucleus of the cells with. And the second box essentially is our fluorescent dye being fluorescent dye you can see underneath that being encapsulated into the cells. Third being merged together. Now, apologies for the picture on the top kind of being a little bit blurry. That picture was actually taken inside of one of those long on a model chips, which is the reason why the picture quality is not as good. But now we're able to take much higher qualities. This was a very earlier picture. The bottom, you can see that we're able to take much higher quality pictures. So you can clearly see them on the actual. You can clearly see on the actual pictures that FITC is equally and widespread distributed across all of the A549s, which kind of indicate to us that the unhealthy cell line models are easily uptaking these lipid nanoparticles, whereas the healthy cell line models seem to be resisting a little bit, but still show some uptake. Now, how do we increase cellular uptake? Well, what we can do is we can directly send our lipid nanoparticles to our cells, and for that, we can functionalize them. Essentially, functionalizing our particles allows for targeted drug delivery. And to do this, we coat the surface of our lipid nanoparticle with a peptide or a ligand that directly identifies a surface receptor on our target cell. Some recent research has actually identified that GLP-1 has been seen increased in blood circulation in patients with lung injuries, things like ARDS, for example, which is deep alveolar disease. And there is some belief that it may have anti-inflammatory properties and may be involved in the repair modulation of the alveolar epithelium. And as a result, I'm kind of trying to target my lipid nanoparticles to GLP-1R. Although that targeting my lipid nanoparticles to target GLP-1 receptors, although there is very limited research on how GLP-1 is expressed on our specific cell lines. We have done some experiments in our lab and we can see them. So they do exist on our cell lines. It's not completely shooting into the dark. And to target my lipid nanoparticle, I'm going to be using exendin-4, a naturally occurring agonist for GLP-1. It's extracted from a very ugly looking lizard, in my opinion, and I'm going to be using it. I'm going to be using it to functionalize my lipid nanoparticles. This part onwards is kind of hypothetical, it's kind of the next stages. But to functionalize, we need to create a new chip. And as I mentioned, every time we create a new chip, we need to make sure we characterize it. That's dimensional characterization, COMSOL simulations, make sure that the chip is operating how we expect it to operate. So one of the first things we did was we essentially added an aqueous solvent, and we pushed just blank aqueous solvents through our microfluidic device, and we found out that our DLS results and nano-FCM results are showing that we're achieving nanoparticles that are in the desired range, show low batch-to-batch variation with a relatively low PDI, and a higher concentration at higher flow rate ratios. This, in summary to everyone, all it means is that this chip is producing lipid nanoparticles like the previous chip without the third inlet. And so the third inlet doesn't have an impairment on lipid nanoparticle formation. So the next steps will essentially be to expose and introduce the lipids and ligands into it. Quickly just touching up on the last part. This is the alveolus on a chip model. As you can see, we've done all of our simulations, and on the top right-hand side, we have managed to achieve 100% confluence on these cells now. There you can see a live dead assay. And the next steps will essentially be to functionalize our lipid nanoparticle with exendin-4. And that will hopefully allow us to generate a pegylated GLP-1R targeted lipid nanoparticle. Thank you very much for listening. Any questions? Thank you, guys. I'm shaking.
Very well. Very clear.
Thank you. Thank you.
Got time for one or two quick questions.
Everyone wants to go.
I have a one which is maybe basic one, but you said about one of the lipids in your cocktail gives you a net negative charge.
Yeah.
Is there a reason why you went for a negative charge rather than a positive charge?
Oh, that's actually a very good question. I'll be honest with you guys. I do not. I think I have an idea. It's because the epoxy not only gives a negative charge, but it also reduces the transient temperature. So it's kind of one lipid that tackles both of the issues. And the reason that we want to lower the transition temperature of the lipids is because otherwise we'd have to do the experiments in really high temperatures. And adding these lipids both makes it so that the nanoparticles at the end repel each other, but also you're lowering the experiment to a room temperature stage. Thank you very much.
Okay, great. Final call for questions. Go on.
Is it, what's the limitation on the range of the types of molecules that could be your drugs? Do they need to be?
Okay, so that's also a very good question. So naturally, because it's a lipid nanoparticle, you have a hydrophilic center core and a hydrophobic tail-like area where the two heads. So between the bilayers, you obviously have a region where you will have your hydrophobic drugs, and inside of the core of the lipid nanoparticle is where you will have the hydrophilic drugs. I feel like because the core is generally larger than the area in between the bilayer, you'll be able to fit more hydrophilic drugs into the lipid nanoparticle. However, drug loading hasn't been something that we've experimented with yet. And I guess I'll have to experiment with both hydrophobic and hydrophilic drugs to see how my nanoparticle is performing. Does that answer your question? Thank you.
And we've got one last quick question just behind Cam.
It's all right.
Thank you for that. That was really good. So in terms of stability, so obviously you're going to load the drugs. Do you know how you're going to store them? You're going to freeze dry them, and does that affect the lipid stability?
Right. So one of the things I've actually been looking since I started this was how can I store my lipids in a safe way for a long period of time? And everyone is always telling me about drying them. But everything that I'm looking that involves drying them means that you're losing a lot of lipids because they break apart. So at the moment, I'm not until I figure out exactly how to increase the survivability of, like, I guess, my lipids or my liposomes. I won't bother with things like freeze drying because right now it's just breaking apart the lipids. So the long-term storability is still a question. I do ideally want to concentrate these lipid nanoparticles so I can have like one drop that has 1 billion particles. But at the moment, I'm producing the order of millions. And I do need to find a way to concentrate them. Currently, I don't have a working method.
Well, thank you.
Worries?
Excellent. Okay, we give a final round of applause to all of our